is Emma Winger, and this is my video for Lab Activity 1 about G-coupled protein, or about G-protein coupled receptors. Today I will be discussing G-protein coupled receptors, which can be abbreviated GPCRs, and we'll be talking about what they are, what they do, and the different subunits, with a focus on the alpha sub S and the alpha sub Q over 11 subunits. G-protein coupled receptors. So what they are. They are receptors activated by extracellular chemical messengers that result in a second messenger being released within the cell. They are particularly important to water-soluble molecules, as these are polar and cannot diffuse through the cell membrane, which is nonpolar because like dissolves like. So cell membrane is not permeable to molecules such as water-soluble hormones. An example of a water-soluble hormone includes epinephrine, which through a G-protein coupled receptor protein leads to the inhibition of glycogen synthesis and the promotion of glycogen breakdown. But we will get into the details of some G-protein coupled receptor pathways later. G-protein coupled receptors. Now let's talk about what they do. As we stated, they result in the release of a second messenger within the cell. Let's illustrate this process. Here we have our cell's plasma membrane with a receptor sticking out. This receptor is coupled with a G protein, hence G-protein coupled receptor. An extracellular messenger, known as the first messenger, binds to the receptor and it never actually enters the cell. This is significant to note because, as mentioned earlier, it doesn't really work out for polar molecules to cross the nonpolar membranes of the cell. This mechanism is important to convey the signals that we need to get across the cell and to get our desired response. When an extracellular messenger binds, the G protein can then activate the effector protein. We will get into examples of these later. The effector protein is then able to produce that second messenger. And this second messenger often is involved in activating protein kinases, which, through a series of steps, can lead to a desired response in the cell. So that was a pretty vague illustration, but we'll continue to get more in depth as we go. Now let's talk about the subunits. So the G-protein coupled receptor has three monomers, the alpha, beta, and gamma subunits. The alpha subunit can come in different forms, alpha sub S, alpha sub Q over 11, alpha sub I, and alpha sub 12 over 13. We'll be focusing on the two that I've starred here, alpha sub S and alpha sub Q over 11. So let's first take a look at alpha sub S and mention the key players in this mechanism. So the key players would be cyclic AMP, which is known as CAMP, which is the second messenger of this pathway. We also have adenylyl cyclase, which can be abbreviated AC, and that's the effector protein in this. And then we also have protein kinase A, which is abbreviated pKa. So when the first messenger, or hormone, binds to the receptor, the G protein with subunit alpha sub S is activated, and this does also require for GTP to be replaced by the higher energy version of itself, GTP. This active G protein is then able to activate adenylyl cyclase, which uses some ATP to produce the second messenger CAMP. CAMP is then able to activate protein kinase, which then triggers the desired response. This desired response might be activating enzymes, stimulating cellular secretion, or opening ion channels. Now let's take a look at alpha sub Q over 11. The key players in this include IP3, I'm not even going to try to say the full out name, and diacylglycerol, which is DAG for short. And these are second messengers. We also have calcium, or calcium 2+, which is sometimes referred to as a second messenger. We also have the effector protein, which is phospholipase C, abbreviated PLC. And we have the membrane protein, which is also a bit too hard for me to say, but we'll call it PIP2 for short. And this is a membrane protein, as mentioned. And then PKC is involved in this, which is protein kinase C. So when the first messenger binds to the receptor, the G protein with subunit alpha sub Q is activated. The activation of the subunit again requires for GDP to be replaced for the higher energy version of the molecule, GTP, which is going to kick off that GDP when it replaces it. This active G protein activates phospholipase C. Phospholipase C then cleaves the membrane phospholipid known as PIP2, right here. And PIP2 is cleaved into DAG, diacylglycerol, and the molecule IP3. DAG will remain associated with the cell membrane, but it's going to be involved in activating PKC. And then PKC, or protein kinase C, can then activate other cytosolic proteins, resulting in the desired cellular response. IP3, on the other hand, can also lead to a desired cellular response. It first travels to the endoplasmic reticulum, where it will bind with a receptor, a channel. It'll help open a channel in order for calcium 2+ to leak out of the lumen of the endoplasmic reticulum. And then through a series of steps going on, it can eventually also help lead to the desired cell response. This desired cellular response could include responses such as contraction or secretion. And that is a brief overview of G protein coupled receptors and what they are, what they do, and the significance and differences between some of their subunits. Thank you.